Okay, let's look at Salmonella. Just quickly now, as we discussed earlier, so you'll have the epidemiologic setting, you'll have, in this case, is a farm, it could be a hospital, it could be environment or others. So we have a specific epidemiological study design. In this case was also a cohort, with controlled intervention study, but it's a longitudinal study. To 3 years, certain number of barns, I don't have all that the details, but dedicatively using different classes of disinfectants to be able to see its association and selection for antibiotic resistance. So the most direct question is, does the disinfection process in these swine barns work? And we expect that it would work. So here is what it shows you. On the x-axis, you see 4 different disinfectants. Biosentry, which is a quaternary ammonium compound. Synergize, has a QAC, quaternary ammonium compound with glutaraldehyde. Vercanis, it's a peroxygen blend. And then we have a controlled water. And this is from 36 barns in North Carolina, but recently done after I moved to Ohio. So if you look at the proportion of Salmonella, all these 3 disinfectants reduce the proportion of Salmonella. The prevalence is lower. They didn't get rid of it completely. It's not surprising, but they reduced, which is encouraging. Biosentry, all of them, they work okay. Another interesting fact we saw is the control. If you see before disinfection, what you see in blue, the control barn had maybe about 15% positive for Salmonella. After it's not really disinfection, but hot water wash, in fact, it looks like it spreads the Salmonella much better. We had a higher proportion, more than 20%. So in fact, the advice, if a farmer asks me, should I just use water? From Salmonella standpoint, either you better disinfect or not just use water, some other means. But definitely that was some simple observation we had. Now, look at those isolates, the surviving bacteria after disinfection. Do their antimicrobial resistance profile change? And to do just this level, I don't need a molecular thing, but this is important information. Phenotyping is also very important. So this is an example I give you from Synergize. That was the largest extent. All of them are, but looking at pre-disinfection and post-disinfection. But look at after disinfection, the isolates that we collected after disinfection in testing the x-axis for different antimicrobials, that the proportion of resistance went up. That's a phenotypic level. So at least we learned that, okay, disinfection reduces Salmonella, but the surviving ones tend to be more multi-drug resistant. And we looked at, by the way, this was not my interest at all, but as we were discussing with one of our collaborators, John Gunn, who does Salmonella pathogenesis work, he was interested to see virulence, and I was asking him, we see, this, based on PFGE fingerprinting, they look identical strains. We collected them from the same barn, the same pen, or where the pigs are staying, but before disinfection and after disinfection, the antibiotic resistance profile changed. Maybe in some of them, we look at descriptively, it had just tetracycline resistance. After disinfection, on fingerprints, they look the same, but their phenotype changed to highly multi-drug resistant, penta-resistant, hexa-resistant. And we were wondering, how about virulence? Did virulence change? So we blinded John in his lab, and we gave him some isolates, before and after. Like these 2 are before and after, these 2 are before and after, and this is another after from the same pen. So he found first just looking at invasion assay, okay, cell invasion assay. I'm sure Dr. Toshoma does lots of assays, or maybe, that he found that the pre-disinfection one was not able to invade much, but statistically significantly we had number of replicates and more isolates we gave him. Just for the sake of demonstration, I picked one interesting one, that the cell invasion ability went up after disinfection. So there must be some other functional consequences on the organism. So the environment driver being the disinfectant, the pathogen aspect is the changes in antimicrobial resistance and changes in disinfection. he was then interested to look at, he asked, should we do macrophage survival assay? I said, absolutely, go for it, let's see. On macrophage survival assay, he found the same thing. Their ability to survive was higher than after disinfection. Isolates after disinfection, again, the same DNA fingerprint. The genotype looks the same. Functionally, something changed. And then he proceeded doing mice inoculation and survival of mice. And at least the this particular strain 12136 killed all mice by day, by day 5, whereas the pre-disinfection, the ones which received the pre-disinfection isolates from disinfection, pre-disinfection survived. Okay. So clearly, in fact, maybe about this level, after he did the first 2 assays, we kind of discussed the details. We blinded his lab up to that level and discussed this is the story of the study. So in fact, he's doing now more assays, doing whole genome sequencing to see is there a genetic difference, is there plasmid loss, is there a functional gene expression assay. We are in that process right now. But clearly, the use of disinfectants selects for that. Oh, I thought I didn't have Acinetobacter baumannii, but definitely I think I have some of this. We did lots of study also in Acinetobacter baumannii. Since this is not PowerPoint, you might not be able to see it, but comparing the different disinfectants and their ability to form biofilm, their changes in antimicrobial resistance. Just quickly to tell you, again, Acinetobacter baumannii is one of the big ones. This is all isolates from humans that originated from the from human patients from our medical center, and they had our clinicians, IDs, specialists, epidemiologists, infectious disease epidemiologists had issues with having this highly multi-drug resistant Acinetobacter. So we did a study for 2-3 years. I had 2 postdocs working on a few projects on that, but what we found was definitely the disinfectant types they use in their wards had a lot to do with the, in fact, what happens is not like just a selective pressure, but the resident Acinetobacter expresses these highly multi-drug resistant efflux pumps when they get exposed to some disinfectants. That has also been, Laura Pidot, others in UK and other places did a study in Salmonella and others, but we characterized a number of different multi-drug efflux genes that triggers multi-drug resistance when they have this substrate of disinfectants exposed to that. So I'll skip when you expose one. And then other co-selection, we have been studying the tolerance to heavy metal micronutrients. These are used in animals as, to some extent to control some infections. At least zinc is known to do that, but for various purposes, just to promote better production, productivity of the animals, and other factors, which is good, in a way from the producer's standpoint. Scientifically, it could be a challenging thing. PCO is the operon. Operon is a set of genes. I think we have it on the terminology definition on the day 1, but it's a PCO is that operon is also a multi-drug efflux system. So what bacteria does is once the copper ions that are in the feed consumed by the pigs and gets exposed to this salmonella, it can move through the cell membrane into the cytoplasm, but the PCO operon will pump it out. It's a flux system, pumps out quickly. But once that operon is activated, does it also pump out antibiotics? Is the question we are interested. Because having this multi-drug resistant salmonella will be an issue to human health as we consume food or the pork products and others. So this is a fairly large study, but I will give you quickly. So, is there a copper tolerance among the salmonella isolates? Yes. That was, we had a good proportion, but again, I'm showing you some of the key slides. Is that copper tolerance associated with multi-drug resistance? So you will see here in the columns MDR and non-MDR in terms of antibiotic resistance salmonella, and those isolates with low MIC4 copper, that's less than 20 millimolar, or those with high So we took 20 as the breakpoint, more than 20. So if you look at the odds of having a multi-drug resistant salmonella, when you have that isolate is also copper tolerant, is 3.3 times more than the odds of having a low MIC to copper and no multi-drug resistance. Is that clear? Okay. So there is association between heavy metal use and multi-drug resistance. You follow me okay so far? Okay. And then we looked at within the multi-drug resistance, within this set of isolates, there are different penta-resistance patterns we commonly see. Is there a specific MDR association? Because we characterize this and published a lot on this, mainly looking at whether what genes, where are they located, that sort of thing. Even though their pattern is similar, ampicillin chloramphenicol, there is the other one has a kanamycin, but their genetic background is also different. Many of the genes are different. So what we found was, again, there was a strong association between one of the resistance types, but not the other. So there was distinct MDR multi-drug resistant isolates were selected when we do that. And from molecular standpoint, there's PCO and other genes we've been looking at. So what happens at the pathogen level, multi-drug efflux pump, there are different operants acting for that in Acinetobacter, in Salmonella, and various other ones. But the problem is, even when you use non-antibiotics, substrates that can not just select the strains, but trigger the gene expression. We looked at again the first day, and then translation, the functional, highly effective multi-drug efflux pump. Some of them are non-specific. Once it is tolerant to copper, it can pump out different class of antibiotics. Just because of you have a set of these operants and functionally active, they can be resistant to so many different classes of antibiotics. Okay, so far? So, okay, I'm getting back. So what are these elements causing Salmonella to become multi-drug resistant? Here is what I was getting into, the class 1 integrons. So what you see here is a typical architecture of class 1 integron. They have an integrase, a gene that serves as a hotspot to antibiotic resistance or toxin genes. And they do site-specific recombination, and after it got inserted. And these integrons necessarily always, class 1 integrons particularly, carry quaternary ammonium compound resistance gene. It's a deletion but reduced susceptibility to quaternary ammonium compound, and sulfamethoxazole resistance gene necessarily, always. Okay, this is just a general aspect of what integrons look like. And this is from our previous study, but looking at Salmonella, different serotypes, you can have this integron profiling. In fact, if they are the same serotype, you can use it as to differentiate strains, but you have other choices. But one strain can have 2 or 3 different size integrons. They don't just carry one integron. And these integrons can trap not just one gene, several genes. There has been in Vibrio, in fact, about maybe 10 years ago, super integrons, they called it super integron, that served as a hotspot to about 100 genes. Clear so far? Okay. Question? I'm going a little bit too fast than I did this morning, but yes. I wouldn't say not every salmonella is not necessarily present. There are some strains that do not have integron. Sometimes you may get antibiotic resistant one, but it's carried in a different location, not all integrons. But the key thing is integrons are really a key mediators of multi-drug resistance. They carry many genes at a time. Here is an example. This is what you see on the top. You may not be able to read all the details of the genes, but what I show here in a darker arrow, the white arrow, is 2 integrons within a 13 kilobase pair region chromosomal location of the most common multi-drug resistant Salmonella serovar-typhimurium. There are two integrons. One of them carries adenyl transferase, which encodes for streptomycin resistance. The other one carries beta-lactamase, encodes for what? We discussed this morning, earlier. Penicillin, ampicillin, beta-lactam categories, amoxicillin. And integron, as I showed you, already has a quaternary ammonium compound and sulfa-methoxazole resistance, both of them. But in the intervening region, there is a tetracycline resistance and chloramphenicol resistance. Okay? So within this 13 kilobase pair region, you have all these 5 resistance genes clustered together. It's important information if you are doing this study, but I'm giving you example from our studies. In one of our studies, this is a small kind of dendrogram. You remember we discussed dendrograms, everything in phylogeny yesterday. You see this 100% similar isolates, right? Do you see that straight line? Here, yeah? Remember all that dendrogram, phylogenetic discussion? But phenotypically, we notice some of them are multi-drug penta-resistant, some of them has only sulfa-methoxazole and streptomycin resistance. But on fingerprints, they look the same. So again, DNA fingerprinting, if you do like pulse and field gel electrophoresis, it indirectly tells you the presence of sequence variation, but they are not high resolution methods. If you are really focusing on specific area of interest, toxins, antibiotic resistance, virulence, you have to zoom into the gene. So now we are really zooming into part of the chromosome. So what we found was part of this genetic architecture between this and this region was deleted. It looks like it recombined. And they had those, other than that, they look genetically the same, genotypically identical on fingerprint, but these strains with only sulfa-methoxazole and streptomycin had only 1 kb integron, one integron of 1 kilobase per size. Whereas the penta-resistant ones, which we commonly found from the GI tract, had this highly genetic architecture. Maybe, again, the selective pressure. Maybe when they get into the environment, they don't need to carry tetracycline resistance gene, chloramphenicol resistance gene. It looks like there is also a conserved segment. You may not be able to see it, but this little 54 base pair region here is conserved segments that they pop out that middle segment. They recombine and pop out that segment. So whereas they are penta-resistance here, they're just sulfide. So this is, after successive, previous study I mentioned to you, looking at conventional farms, antibiotic farms, we have seen some variation resistance. Genotypically, some of them look identical. So also this enables you to appreciate the value of phenotyping. Okay, as discussed, just because you did a cool, nice fingerprint molecular method, it doesn't mean that you have to forget the phenotyping. It could really supplement with what you find with the genotype. Yes. Serotype is one. It is one serotype. They are Salmonella. Yeah. Good question. Very good question. So how come? So maybe somebody can help on this. How come? So the question is, how can they be genotypically similar while they lose this part of the genome? Can anybody help answer? Why do they look similar? Do you expect them to be similar? Okay, let's just revise a little bit. And let's assume this is a whole genome of Salmonella. We discussed a little bit some of the genome sizes. Can anybody say what's the genome size of Salmonella? Okay, that's okay. E. coli, Salmonella, Campylobacter, mutations. But anyway, about 4.5 megabase pairs, not megabytes, okay, 4.5 megabase pairs. Okay, when we do PFGE, what do you use? Okay, tell me pulse field electrophoresis, DNA fingerprinting. What is the method? What does it involve? Loud? Okay, there are core methods. Do we do amplification? Yes or no? No. Okay. Do we do restriction? Yes, correct. What kind of restriction enzymes do we use? Rare cutters. Great. So if you do rare cutters, you may have, let's see, maybe it cuts it about, I don't know, 37 to 40 places. Okay. These are large fragments that we are separating on unique type of electrophoresis chamber, right? Contour-clamped homogeneous electric field. But now let's look at what is now we zoomed in, as I said earlier. Now we are investigating this particular multi-drug resistant strains. So how big is that multi-drug resistance architecture I just showed you? How big did I say? 13 kb. So that 13 kb might be, this size, right? It's a very small part of the whole genome. You following everybody? That's a very good question. So when you do PFG, you remember the high resolution methods, not so high resolution. PFG is not that high resolution method, unlike maybe AFLP. If it's a small difference, it doesn't show you because you get these large fragments. Even if this large fragment is 13 kb different from the next one, they may just look identical, just like that. That's, technically that's the reason. Okay? Very good.
And then you use the same technique to analyze the properties and FG can use to zoom in.
Yeah, you can do that. There is possibilities. You can, depending on the size. Especially the lower it gets into, like, let's say 50 kb, you can cut out that gel, purify it, you can do some plasmid, or you can, preferably, you can use other methods. AFLP and others where it gives you more higher resolution, but more number of fragments, more technically demanding, but you can do many things. Okay, last slide. So just to summarize MDR salmonella, again, we didn't get into the PFG other parts, but the key thing is, so this is being an example. You might be studying different things, but you have to understand the pathogen. In this case, we looked at the pathogen and the environment aspects, environmental pressure. You might be studying the host factor and the pathogen. You might be studying the pathogen environment or the triad dynamics. In this case, our main interest is antibiotic use and resistance, or the non-antibiotic co-selection and resistance. But you have to have a very good understanding of the molecular components to be able to answer. This is a very truncated presentation as compared to the whole study, showing the PFG, the clustering, the different strain types, the phenotypic relatedness, then looking at integrons. It can go more in detail. I think with your presentation that is uploaded on the online system, you'll see a larger, a broader, detailed presentation. Okay? Any other question on MDR and antibiotics, but not the antibiotics that politics. I think we spent enough time discussing this. Okay. What? Alcohol, ethanol, or I'm not aware of that. I'm not aware of any. That's a wonderful thing. Definitely, alcohol is properly, and I'm not aware of it, but many substrates, even they are not disinfectants. Others, TBM, and many others could serve as substrates to activate. So, once the pathogen is exposed at sub-inhibitory concentrations, they can activate these multi-drug efflux pumps. That's a whole another big area of study. Other questions? Okay. Later on, we'll come back to Clostridium difficile and MRSA, just mainly showing you, what we discussed, the use of MLST, the use of PFG in addressing specific epidemiological questions. So we'll end our salmonella on this